when I get the lovely 30-second delay, which is always fun to experience. Ooh, that's going. All right, we are in the right spot.
Good deal.
Excellent. All right. Oh, Helena?
Yep.
I need to make you co-host. There we go.
Thank you.
That way, if something horrible happens and I drop out, you're right there.
Well, that's what I'm here for,
There will
support.
be no problems at all. So.
My internet at my house today was not cooperating and I was pretty nervous. I think I got it under control though.
That's great. That's great. Well,
went out at NAU for about an hour. Was that just today or yesterday?
Oh gosh.
They all run together, but that was fun.
It always seems to happen during a live stream or a big presentation or something. Something goes horribly wrong, and yeah, we've had that happen more than once.
It all works out in the end.
Yeah, I mean, it all gets done. Yeah. Luckily, we're not doing heart surgery at the moment, so you know, everybody will survive and be okay. Emily, I'll put the spotlight on you when you start talking. So just in case, if we chime in with anything, it won't be our faces. It will still be your face for the whole time. I'm sure everyone will appreciate that. So, and it looks like we're getting some participants coming in. So that's good. For those of you out there joining us, we'll be starting the program straight up at 6:30. So hold tight. We'll have some housekeeping and then we'll get going.
Well, I'm really excited for this. I think it's going to be a great talk.
I think it'll be fun. I have to give credit to my friend and colleague, Bridget Barker at PMI for coming up with the title. We were working on a panel for something else. So I cannot claim any creativity with that, but she let me use it, so.
Well, it certainly attracted me when we were all offered, you know, the list of different programs to host and everything. I was like, I'm going for the Snot one. How could you not? I think a lot of people will be attracted to it. So looking forward to it.
Talk microbiomes.
How did you get started? What interested you in microbiomes in the first place?
Snot.
It's not any of your business, Sherry.
Apparently.
But, we met a ENT doc at a meeting my PhD advisor and I did, and just got talking to him about chronic sinus infections and all of that stuff, and he led us into his OR, and I nearly passed out. It was pretty gruesome sinus surgery. I did not expect that. So
oh wow.
I stuck to the science side of it. You can send me the snot and I will look at that. But
you don't want it coming out of people or anything like that. No, I get that. Makes perfect sense. Well, I'm glad you it's probably not something that immediately jumps to people's minds. I'm going to study mucus. That would be a great thing to do. So I'm glad you took that on and are furthering your research. It's a marvelous thing. All right. Oh, up to about 14 participants now. That's good. We usually gain a few in the first 5 minutes once things get started. So looking forward to some good questions as we go along. And I'm stunned that I'm already cold again. I had to put on a big sweater to start this.
Flagstaff fall is the best. It's heaters in the morning and air conditioning in the afternoon.
Crazy, crazy. All right, well, it is 6:30, so I am going to promptly start. Thank you very much, all those out there, for attending It's Not Trivial, the role of the human microbiome in respiratory disease. My name is Sherry Schaefer, and I am a member of the Flagstaff Festival of Science Board. Joining me are fellow board member Helena Murray and this evening's presenter, Dr. Emily Cope. But before we start, I do have a little bit of housekeeping. First, you need to know that the festival is live streaming this webinar to YouTube, and only the hosts and panelists will be visible or heard during this simulcast. So if you miss anything or you want to see any of the other live stream programming, including a lot from last year, just go to our YouTube channel. This event is part of the Flagstaff Festival of Science, often called the best 10 days of the year. Our mission is to connect and inspire the citizens of Northern Arizona, particularly the youth, with the wonders of science and the joy of scientific discovery. Your participation and support are vital to our shared success. All the festival events are free, thanks to our generous sponsors and donors. If you would like to support our work, please go to scifest.org and click donate. You can see a full list of events at scifest.org, and you can register for any of the events that require it on our Eventbrite page, which is flgscifest.eventbrite.com, and there are links to the festival's website and Eventbrite. I'm doing really well. Eventbrite page will also be in the chat. We'll put them in there after the presentation starts. One way you can support the festival is to take a short 5-minute survey after completion of this webinar and any other events you participate in. The data is really helpful for us to help make the festival better each year. The survey can be found in the chat. I'm going to post that. And also, one survey respondent will win a pair of Apple AirPods after completion of the festival. So the more things you go to and the more surveys you complete, the more chances you get to win. During the presentation, and this is really important, please type your questions in the Q&A box. The chat is only going to be for any technical problems you might have. So go to the Q&A box. It'll also allow you to upvote questions. So maybe you see something that you already want to know, go ahead and hit upvote. Due to possible time constraints, we may prioritize those that get more votes. Thank you so much for patiently waiting through the housekeeping portion. I bet you're pretty excited to get going. I know I am. So without further ado, Dr. Emily Cope is an assistant professor in biological sciences and assistant director at the Pathogen and Microbiome Institute. She earned her PhD here at NAU, which was followed by a postdoctoral research fellowship at the University of California, San Francisco, with Drs. Susan Lynch, Andrew Goldberg, and Steve Fletcher. She has a broad interest in host microbe interactions and in chronic and progressive disease. Thank you so much, and take it away, Dr. Emily Cope.
Thank you for that lovely introduction. I want to thank the Flagstaff Festival of Science, as well as the Office for the Vice President of Research, for inviting me to talk about what we've been up to here at NAU. So I've been here as faculty since about 2016. Time flies a little bit. And I'm excited to share with you some of our research findings and some of our research interests. So as I have learned in the Zoom world, I narrate everything I do. So I am going to go ahead and share my screen and start my presentation. So, as I mentioned, maybe before everyone joined, I have to thank my friend and colleague, Dr. Bridget Barker, also at the PMI, for this title. I cannot claim creativity. We were working on a panel and I just thought we have to use this somewhere. So I'm going to talk about the role of the microbiome in respiratory disease. And I'm also going to I'll introduce you to the human microbiome before we get into some of our research. So if I were to ask you to picture a human, I'm going to open my chat box just in case there are issues, just so I can keep note of that. So if I were to ask you to picture a human, your partner, your child, your best friend, someone you don't like very much, what would you see? You'd probably see something sort of generic like this. Person covered in the skin, our largest organ, typical facial features, eyes, lips, ears, but really, we're much more exciting than that. This is because we're covered inside and out with microbes or microscopic organisms, and these include bacteria, shown up here, fungi, viruses. These also include microscopic parasites. So in your eyebrows actually live tiny insects called Demodex, and they like to live in your eyebrows and eyelashes. So if you need a nightmare tonight, go ahead and Google Demodex eyebrow. But also archaea and in other microscopic organisms. So we really are covered in these things. And indeed, the human body is made up of as many bacterial or microbial cells as we are of human cells. So here we see we're made up of about 39 trillion human cells and about 50 trillion microbial cells. Now that actually drops every time you take a bowel movement, but we're about 1 to 1. Perhaps more impressively, our microbiomes contribute about 100 times more genes to our cells than our human genomes. The microbes in the gut help to educate and train our immune system, break down dietary components such as fibers, and synthesize important molecules such as vitamin K and vitamin B12. And what we'll talk about today is the role that the microbes in the gut might play in peripheral organs. Specifically, we'll focus on the airways, but my lab is also interested in the role of the gut microbiome in neurodegenerative disease. Maybe we'll talk about that at another science festival. So as I mentioned, we're covered in nearly every body site that's been looked at with microbes. Our microbiomes are incredibly diverse and dynamic, and they interact and communicate with each other as well as us, the host. This figure is showing the composition of bacteria that are present on various body sites in and on a person. Some body sites, like the gut, here indicated by this pie chart, are very nutrient rich. They're like microbial rainforests. We have a lot of nutrients. We give our gut microbes nutrients every time we ingest anything from food to our medicines, anything we take by mouth, our gut microbes have first dibs on. So this is also where the highest density of microbes reside. Other body sites, like the skin, are kind of like microbial deserts. They're dry, relatively nutrient poor, so really only the tough microbes that can survive these conditions live here. That is to say, our body sites have different chemistries, different body sites have different chemistries, nutrients, as well as stressors. So each body site harbors a distinct microbiome. But what if we looked across individuals? Would my skin microbiome look more like any other body site on my own body because it's all in my body? Or would my skin microbiome look more similar to your skin microbiome? The Human Microbiome Project Consortium, this is funded by the National Institutes of Health, did just that. They had hundreds of healthy volunteers and they sampled different body sites. So this plot, and we'll walk through it in a minute, so if this looks a little bit overwhelming right now, don't worry too much. We have taken a very high dimensional data set, so all of these comparisons to one another, and we've reduced it to 2 dimensions so that we can look for patterns in the microbiome composition across people and across body sites. So let's walk through this. Each shape represents the microbiome from one body site from one person. The distance between 2 shapes here, so these 2, would represent how similar or show how similar or how dissimilar the microbiome compositions are between those 2 shapes. The other component of this plot is color. So each shape is colored by body site, and don't worry about that list of anatomical names quite yet. I'm going to define them in a minute. One pattern that we can see is that the shapes in each color cluster or group together, indicating more similar microbiomes by body site. So for example, these oranges and reds at the top represent the microbiomes from people's noses, the back of their ear, and their elbow crease. And they look more similar to each other than these other body sites. We can say that as well about the oral microbiomes. So all of the different shades of blue represent different sites in the mouth, and they look more like an oral microbiome than they do, say, to our noses. The green represents the microbiome of the vaginal canal, and of course, brown is poop. So to answer my previous question, it seems like my skin microbiome and your skin microbiome are more similar to each other than, say, my skin microbiome here and my fecal microbiome. And I'd probably say that's a good thing. The other thing we can notice from this plot, that even within a body site, so let's look at stool here, there is some variability in the microbiomes across healthy individuals. So we can ask as researchers whether this variability is important in health status, either current or future, risk for disease, or whether we can change the microbial composition within a body site by changing our habits, like what we eat, how much we exercise, sleep patterns, travel, where we live, etc. And in fact, researchers have shown that geography does impact the composition of the microbiota. In this study, researchers analyzed the gut microbiomes of people from the US here in blue, and also less developed countries, specifically Malawi and Venezuela, here indicated by Amerindians. And this is the same type of plot. The closer two spheres are to each other, the more similar their microbiomes are. And the farther apart they are, the more dissimilar or different they are. So they showed that the gut microbiome of US adults looks very different than the gut microbiome of individuals of Malawians and Amerindians, despite the latter two even residing on completely different continents. They also showed that not only are the bacterial taxa different in the US gut microbiome, but we also have fewer, I'm using the word different a lot, fewer different types of bacterial taxa, AKA lower diversity. So this plot on the x-axis is showing the number of different types or species of bacteria in the gut. We call these, in this case, OTUs. And we can see in the US, adults have fewer OTUs or different types of taxa in the gut than Amerindians and Malawians. What could this be due to? Well, it could be due to diet. A typical US diet is rich in protein, whereas diets in Malawi and Amerindian populations are typically dominated by corn and cassava. So what this is suggesting to us is that differences in the number and types of microbes we have in our gut could be driven by long-term differences in environmental exposures such as diet. But the impact of diet on the gut microbiome actually occurs very rapidly. In this study, the researchers took 10 volunteers and they asked them to eat 2 different diets, and we'll walk through this plot too. They asked them to eat a plant-based diet and then go back to their normal diet, and then eat an animal-based diet and then go back to their normal diet. So on the x-axis is time by day. So this is individuals eating a plant-based diet. That's just what they were eating normally. It could be like 10 pounds of chicken nuggets or all quinoa. And then here in the shaded area are when the individuals were asked to be on this very specific diet. And so on the y-axis, we're looking at another measure of microbial dissimilarity. And so what we're asking is, each individual's microbiome change from the baseline level when they're on an individual diet? And so the lower the value is, that means the more similar their microbiomes were to their baseline. And the higher the value is, means that they're more dissimilar. And so, I also wanted to point out these blue arrows. The blue arrows are tracking dyes that they put in the diets. And that allows us to see essentially when the food reached the GI tract and then when it was out of their system. So when we look at individuals who were on a normal diet and then started their plant-based diet, it didn't do too much to change the composition of the microbiome or the types of bacteria that were there compared to baseline. Though later in the paper, we do see that the products or metabolites were quite different. But interestingly, we see a very rapid change one day after the food reached the GI tract of these volunteers' gut microbiomes when they were switched to an animal-based diet. So does diet have an impact on host health? We know, based on those 2 studies I showed you, that it influences the microbiome composition and function. So what bearing could this have on our health status? Well, I mentioned that microbes in our gut metabolize the fibers we eat in our diet. In fact, our human cells cannot make use of fiber until it's broken down by the microbes in the gut. And it's broken down to molecules called short-chain fatty acids, which here I'm indicating in the red triangle. And this was the product that I alluded to in the previous slide that was increased in volunteers when they were eating a plant-based diet. So it turns out that these short-chain fatty acids are pretty important. In the gut, they're used as energy sources for our colonocytes, or the cells that make up GI tract. So this is representative of the gut, and these are dietary fibers. And those that aren't used by colonocytes, these cells here, can cross over into the host side of things. And they can inhibit certain types of inflammatory cells. And so that's what I'm showing here. They can also, short-chain fatty acids have been shown to increase the number of anti-inflammatory immune cells called regulatory T cells in the gut. But these don't just interact with our guts. They can make it into circulation, so they can reach the blood, where they can also travel to other organs. And in the end, they can inhibit inflammatory immune cells that are causal of our respiratory diseases, such as asthma and chronic sinusitis. But in my lab, we don't just study the gut microbiome. We're also interested in the microbes that reside in the upper and lower airways, such as your nostrils, your sinuses, and your lungs, and how these microbiomes differ in disease states compared to healthy states. We know that studies have shown a few important features of the respiratory microbiome in health and disease. Firstly, in health, we have a higher burden or total number of microbes in our upper airways, and the total number of microbes decrease as we reach our lungs. But in a disease state such as asthma or cystic fibrosis, the total number of microbes present in the lungs increase. We don't really see a change in the total number of microbes in the upper airways, but we do see a change in the composition of the microbiome. So in health, our upper airways have a high relative abundance of this red here, or actinobacteria, as well as Firmicutes. And our lungs harbor a microbiome as well. So they have a higher relative abundance of this yellow, which is Bacteroidetes, as well as Firmicutes. So today we're going to talk a little bit about sinusitis and asthma, which I'll define in a couple of minutes. But in airway disease, we see a shift or an alteration in the composition of our microbiomes. So in sinusitis, we might see overrepresentation of some bacteria, such as Firmicutes. And in asthma is interesting. We see that there are more microbes, a higher microbial burden, but we also see a greater diversity of microbes that reside in the lungs. So a lot of the work that we do in my group is to figure out kind of what all this means and whether and how these changes in the microbiomes contribute to disease. So as I was introduced before, the interests of my lab are broadly to understand the microbe-host interactions that contribute to chronic and progressive disease, including the role of the respiratory microbiome in chronic rhinosinusitis, asthma, and cystic fibrosis, as well as the gut microbiome in driving or preventing inflammation in other organs, such as the lungs, the sinuses, and the brain. For the latter, we're specifically interested in the devastating neurodegenerative disease, Alzheimer's disease. But we're not going to talk about that today. We're going to focus on respiratory disease. But rather than let me tell you all about this. I'm going to introduce you to my group and show a short experiment we did to introduce the idea of a microbiome. So this video was directed and produced by PMI's creative genius, creative director, Ken Ng. And I'm just going to show this to you really quickly.
I grew up in a rural part of California, and I wasn't exposed to any university professors. But I love science and nature, and my 3 horses, so I thought I wanted to be a veterinarian. I came to Northern Arizona University as a pre-vet student for my undergraduate degree. I started in a research lab, which set me on a road to a PhD. I didn't know it when I was a kid, but my undergraduate experience at NAU made me realize that my passion is being able to make discoveries and ask interesting questions that can improve human health. I now lead a group of wonderful researchers. Not only is the research side fun, but I also love mentoring students and seeing how far they go in their careers and their lives. It's a dream job. We study how the microbes, or the tiny living things like bacteria, fungi, and viruses, make you healthy. All of the microbes that live in and on you are called the microbiota. Did you know that you have at least as many microbial cells in your body as you have human cells? This makes you at least as much microbe as you are human. We study what happens when our microbiomes are disrupted and how this can lead to some common diseases like asthma, infections, and Alzheimer's disease. We researched these microbes that live in the nose and in the gut by reading their DNA and growing them in the lab. Our goal in the lab is to figure out how to harness the power of the microbiome to improve human health.
I have love-hate relationship with math. When I got into science, more specifically a biology major, I thought that I didn't need it. But when I started work in the lab, math was everywhere. By then, I fell in love with microbiology and had to make peace with numbers to do what I really want to do. And since I'm Korean, I went back to Korea for military duty. I served the Korean Army as a research specialist for 3 years doing microbiology. After the Army, I worked at a global pharmaceutical company as a quality control personnel for vaccine production. The money was good, but there was still something within me that desired more research. So I got into Korean University for a PhD. I returned to PMI as a postdoc to work with brilliant people like Dr. Cope and Dr. Paul Keim. I am now studying a relationship between the airway microbiome and chronic diseases such as cystic fibrosis and chronic rhinosinusitis. My ultimate goal is to win a Nobel Prize and save the world. But for now, I'm looking forward to being a professor at a university and leading my own research lab. Working here at PMI with great scientists is a solid foundation for my future goal.
At around the age 6, I became fascinated in microbes and the diseases they cause. I spent a little more time than the average 6-year-old watching Discovery Health. Because of this, I always thought I wanted to be a doctor, but when I got to college, I realized I wanted to be the one asking the questions instead of diagnosing the patients. I began at PMI in the Cope group as an undergraduate student. I was working on a research project where we looked at the gut microbes in mice with an airway disease. Let's just say I spent a lot of time collecting mouse poop. As a graduate student in a PhD program, I teach anatomy, take classes, and get to work on my very own research project. I'm working on understanding how gut microbes are related to a brain disease that causes elderly folks to lose their memory. Again, a lot of mouse poop. After I earn my doctorate, I would like to work in clinical research to help sick people feel better. Being a scientist and a teacher is something that I've always wanted to do. NAU and PMI has given me that opportunity.
When I was little, I loved science. I collected rocks and plants and science magazines and said, I'm going to be a doctor. But in my world, nobody went into medicine. My sister and I were the first ones in my family to ever try to go to college. I didn't have many role models. Pursuing my childhood dreams combined my love of science with wanting to help people. A friend told me about Dr. Cope's microbiome research lab. I immediately took out my phone and Googled her research. Her lab studies asthma, which is something I grew up with. I had trouble breathing and I couldn't play sports. She also looks into how our diet can affect the microbes in our gut and then affect diseases like asthma. I hustled a meeting with the team and soon joined the lab as a research assistant. LSAMP funded my work on an asthma study where we got to grow different bacteria on agar plates. LSAMP's goal is to help students from diverse backgrounds get involved in STEM. Our work in the lab helps us better understand health and illness, which leads to innovations and treatments that can potentially save lives. I used to take pills and inhalers for my asthma, and now we can possibly find treatments that can replace those medications. I dreamed of being a doctor, and now I'm working towards becoming a pediatrician. I will remember PMI when I hand a 3-year-old a lollipop in my future pediatric practice.
When I was younger, my father would often take me to work with him and show me a lot of cool things inside of one of our local hospitals. He would also allow me to go into operating rooms where surgeons would be performing life-saving procedures. He once pulled out a broken microscope and said, "Let's fix this." I didn't know what he meant by this, but I wanted to help. This was my first experience with microbiology. As a child, I was inspired by both doctors and nurses who would tell me about their work and also what they would do to help people feel better. This motivated me to study biology. In high school, my biology teacher recognized my hard work on assignments and urged me to participate in research. I took her advice and I had an opportunity to work on cool engineering projects. One was concentrated solar power and the other was cost-effective hydroponics. These small projects turned out to be a big deal. At NAU, I learned about the COPE lab after my biology professor recommended that I look for potential mentors. When I reached out to Dr. Cope, she was enthusiastic about the human microbiome and how it influences our health. I was super excited to be working with the COPE lab and be making a difference for those with chronic illnesses around the world.
Growing up, my dad had a skull in his office, and my brother pulled out one of its teeth. That's the household I lived in. My mom is a nurse, and my dad is a medical device engineer who makes implants for people's ears. That's where my love of science came from. When I came to NAU, Dr. Cope was one of my first professors, and I thought her class was so interesting that I decided to major in microbiology. I wanted to learn through hands-on experience, and so I asked Dr. Cope if I could join her lab. I get to work on all kinds of fun projects that help scientists to better understand how all the bacteria, fungi, and viruses that live in mine and your bodies make us sick or help us stay healthy. I even earned a grant that funds my own research project. My favorite part of working in the lab is growing bacteria on special plates. In the future, I want to be a principal investigator of my own lab and teach college microbiology classes. Next year, I'm going to start working towards earning my PhD. I'm so excited to work at PMI while I'm an NAU graduate student.
Here's an experiment on how we grow some of these microbes and what will grow from a child's hand and a dog's paw.
Ooh, I wonder which one is dirtier.
Depends on the child. Bacteria are tiny single-celled organisms that are too small to see without a microscope. They're everywhere. In our hair, our skin, our mouths, dirt, soil, lakes, you name it. Not all bacteria are bad. In fact, many make you healthy.
That's why I drink kombucha.
Sounds good. Bacteria grow by dividing one cell into 2 cells. Then those 2 cells become 4, then 8, well, you get the picture. Some divide really fast, while others take months to grow.
Psh, slowpoke, I learned to divide in a day. 7.2, 8.5. Booyah!
Here, you're watching bacteria grow on a plate that contains lots of food for microbes. What you're seeing, those small dots, those are called colonies. These are visible to the naked eye when they reach about 1 million bacterial cells, and a colony starts from a single bacterial cell.
My mom says I'm 1 in a million.
Yeah, all mothers sort of do. The microorganisms you're seeing grow on this plate came from a child's hand and a dog's paw. Do you see any similarities? Any differences?
4 fingers, duh.
I think they're both 5. Hopefully this experiment shows you that you have many different microbes that live on your body and on the body of your pets. We can use this knowledge to improve health and find new treatments for diseases using microbes.
Okay, so hopefully that showed you that we can culture microorganisms, and we have a lot of microbes, different microbes that live in and on us. And I also want to point out that everything I show you that's coming from our lab is a huge team effort from these wonderful researchers that I get to work with and mentor. So how even do we profile or analyze the microbiome? As it turns out that even with the best techniques, even the ones we showed in the video, which are just standard techniques, we really can't replicate the human body or any mammalian body to support culturing of entire microbiomes. On a Petri dish like the one you see here, we can typically only culture about 1% of the microbes that reside in any given ecological niche, such as the gut or soil or water. However, there are techniques being developed to increase the percentage of microbes that we can culture. In my experience, culturing is kind of an art. And in fact, if you look up ASN, American Society for Microbiology, Petri dish art, some folks have made some really amazing art in their Petri dishes. But instead of culturing these bugs, we can extract their genetic material, or DNA, and read the sequences of the A's, T's, C's, and G's here. And then we can then use these sequences to determine which microbe the DNA came from. But in a complex community like the stool or skin or nose or snot microbiomes, it's really not quite that straightforward. So let's think back to that cute dog and kid. Say we want to compare their gut microbiomes. Well, there are thousands of different species in the poop, so we have to extract all of the DNA. And we tag the DNA with a marker that will allow us to go back bioinformatically and identify whether a given sequence, so this orange came from the kid or the blue came from the dog. So what we do is we take all this DNA and we sequence it. But then we have at the lower end, 25 million or more different sequence reads that like this. And so we need to use bioinformatic tools such as QIIME2, which is developed by Dr. Capareso's group at NAU and the PMI, to separate these sequences into the correct source. So we can bioinformatically say this tag sequence came from the dog and this tag sequence in orange came from the kid. And then we can compare those microbiomes as we've seen previously. So for the rest of this talk, I'm gonna try not to spend too much time, but I'm gonna walk you through two questions that we're interested in the lab. One is, what is the contribution of the respiratory microbiome, so the sinus and the lung microbiome, to chronic rhinosinusitis and asthma? And two is related to those short chain fatty acids in the gut microbiome. Can we modify the gut microbiome to reduce severity of chronic rhinosinusitis or asthma? And you're at this point thinking, what on the heck is chronic rhinosinusitis, which I'm going to say CRS from now on. CRS is a chronic inflammation of the sinonasal cavity. So I don't know if you know our anatomy of our heads, but we actually have 4 pairs of sinuses, which are air-filled pockets in our heads. So we have our maxillary sinuses kind of in your cheek, ethmoid sinuses right where my glasses hit, and I know this because anytime I get sinusitis, it's in my ethmoid sinuses and my glasses hurt my face. We have frontal sinuses kind of in our forehead, and then we have sphenoid sinuses towards the back of our head. And when these get inflamed and filled with fluid that can't drain out of these tiny little holes called ostia, then it hurts, and you can feel it, and it actually decreases people's quality of life quite a bit. So chronic rhinosinusitis, or CRS, is chronic inflammation of what I just described, lasting at least 3 months, which is really miserable. So patients with CRS, and this is fairly common, affecting about 1 in 20 adults, often report worse quality of life than people living with chronic back pain or diabetes. So what does this look like in a human? If you took a camera and stuck it right up someone's nose and then back a little bit, you hopefully look a little bit more like this. So this is the nasal septum separating the 2 nasal cavities. This is a turbinate here, the middle turbinate, which is shown about here in this plot. And this is the middle meatus where our maxillary sinuses drain into. And in a healthy individual, this looks beautiful, just like shiny and not swollen, no pus coming out. But in chronic sinusitis, it can look a little different. So in the clinic, CRS is differentiated into whether patients don't have polyps or do have polyps. So in CRS patients without polyps, You see this inflammation of the middle turbinate. You see pus coming into the middle meatus from the maxillary sinus. And and the maxillary sinus will be occluded a little bit like this. In CRS with polyps, and these have two sort of different etiologies, CRS with polyps have this giant polyp that originates really here in the sinus and then protrudes through the ostium and out into the nasal cavity. You can imagine this is really uncomfortable. Now, both of these types of CRS tend to recur even when patients are managed medically or surgically through endoscopic sinus surgery. So we think that there's something happening that's persistent in the microbiome. We're also interested in asthma. Asthma affects approximately 1 in 13 people, and it's also characterized by inflammation, this time in the lungs. So it's chronic lung inflammation, and it's also characterized by reversible bronchoconstriction. So patients with asthma take this bronchodilator, which relaxes the airways. During an asthma attack, one might experience wheezing, coughing, shortness of breath, or chest tightness. And this is what the airways of someone who's experiencing an asthma attack looks like. So it's inflamed, it's swollen, there's more mucus compared to this nice open airway. Now, in some types of asthma, we see what we call allergic inflammation, and these 3 proteins, these cytokines or immune cell signals, will come up later. And this is characterized by increased expression of IL-4, IL-5, and IL-13. We received NIH funding to study the relationship between chronic rhinosinusitis and asthma, as well as the healthy upper and lower airways. And this is because these 2 diseases frequently, at least in adults, co-occur together, but we don't understand why. We also know it's been reported that medical and surgical management of the upper airways of CRS, specifically those patients with the polyps, improves asthma status. So we are studying the upper and lower airway microbiome in patients with and without asthma and comparing them to healthy individuals. So this is a partnership with this amazing team at UCSF, Doctors Andrew Goldberg, Steve Pletcher, Jose Garola, and Patricia Loftus. And we've characterized so far the microbiome of 38 healthy individuals, 98 patients who have CRS without asthma. And so far, we're collecting more. This is an ongoing study, 28 patients with CRS with asthma. We did a similar study design. So we're looking at the sinuses, specifically the ethmoid sinus, and the lung microbiomes using microbiome sequencing. So we extract the DNA, we make lots of copies of one gene region, sequence it, and analyze it using QIIME2. We want to know what the contribution of the respiratory microbiome is to CRS and asthma. So first we have to look and see if there are any differences in the microbiome. This is a similar plot to what you saw comparing the number of different types of bacteria in the US population compared to Malawian and Amerindians. But in this case, we're looking at the sinuses of healthy individuals and the lung microbiome of healthy individuals compared to the sinus microbiome of CRS patients without asthma and the lung microbiome in CRS patients without asthma, as well as the sinus microbiome of CRS with asthma and the microbiome of CRS and asthma. And across the board, what we see is a reduced diversity or number of different microbes observed in the body site in patients with disease. So patients with CRS without asthma have reduced diversity, and patients with CRS and asthma have reduced diversity, both in the sinuses and lungs. In a previous study, we looked at the upper airway microbiome of CRS patients, and we found that it wasn't just one type of microbial community that patients with chronic rhinosinusitis had. And in fact, there were at least 4 different types of microbial communities associated with CRS patients. And these were dominated by some taxa that were of interest to us. So you might recognize staph. Some of you might recognize Pseudomonas. But what we found out, what we noticed is that these different microbial communities related to different immune responses in the sinuses as well. So here are patients that are dominated with this bug called Corynebacteriaceae. And these patients have an increase in IL-5. These patients also ended up being those with nasal polyps. So what this study found was that 4 different bacterial community colonization patterns conferred distinct inflammatory profiles, so all of the different types of inflammation in CRS patients. So what are we learning about the respiratory microbiome in CRS and asthma? We're seeing that we have decreased diversity or fewer types of microbes in the sinuses and lungs of participants with CRS with and without asthma, and that we have different types of microbes in the sinuses that associate with distinct inflammatory responses. Now, where we're going next is we're trying to define what is the healthy sinus microbiome and how we can develop new ways to change the disease microbiome more towards a healthy state, and is that going to alter someone's disease status towards more of a healthy sinus environment? So the final thing I wanted to talk to you about today is a new study that we're in the process of publishing. Can we modify the gut microbiome to reduce severity of CRS or asthma? And you might be kind of cocking your head at me thinking like, okay, this isn't going to work. Why would we look at the gut microbiome if we're looking at a respiratory disease? But our hypothesis starts here with that dietary supplement, dietary fiber. So we hypothesize that we can give an individual dietary fiber, which is going to modify the gut microbiome and make them produce those short chain fatty acids that earlier I told you can decrease harmful inflammatory responses. We think those short chain fatty acids get into the blood and block immune activation, and that we'll see reduced inflammation in both the upper and lower airways. And this has been shown that this can work in the lungs. This has never been shown that this can work in the sinuses. So asthma prevalence has been steadily increasing over the past 30 years. And we know that this can't solely be attributed to genetics, and it's likely a complex interaction with genetics in the environment. In addition, asthma disproportionately affects individuals who are living in urban or low socioeconomic areas. And we also know that socioeconomic disparities in health behaviors exist in our population, including increased consumption of a low-cost, high-fat, high-sugar Western diet that's also low in dietary fiber. And as we saw in the beginning, diet is a major modifier of the gut microbiome, and kind of good news for us is that this can happen relatively quickly. So our hypothesis is that gut microbial metabolism of this fiber supplement, and you'll see this as SCF, and I'm going to try to clarify on all of our slides, is going to lead to increased short chain fatty acids and decreased upper airway inflammation. So how did we do this? Well, we took a preclinical model using lab mice, and we had 6 groups of lab mice. Group 1 and group 4 were just given PBS, which is just saline, and you could drink it. It's not going to cause anything, any bad reaction. Group 2 and group 5, we gave them an allergen, which is going to induce the IL-4, 5, and 13, and this is called OVA, or ovalbumin. And then in group 3 and group 6, we gave mice that allergen, ovalbumin or OVA, and then also gave them oral dietary fiber. So they just ate, actually they drink it because it's soluble. They drink it in their water. We separated groups into 2 diets. We had a controlled diet. So this is just kind of mouse chow that is healthy for them. And then we developed a Western diet that was very high in fat and also high in sugar. So the control diet had more carbohydrates, so you can kind of equate it to more plant-based, and the Western diet had more fat. Well, let's look at 2 of those genes, IL-4 and IL-13. On the x-axis, we are looking at different mouse groups, those 6 groups. Mice given PBS that were either on a control diet indicated in blue or a Western diet indicated in red. And in both cases, they're not really seeing any allergy. When we give mice ovalbumin, the allergen, we see increases in those 2 genes that are markers for allergy. So IL-4 and IL-13, both in the Western diet and in the control diet fed group. And then when we gave mice fiber plus the allergen, so compare this bar to this bar and this bar to this bar, on the control diet, we see a significant reduction in IL-4 and IL-13 gene expression. But interestingly, we don't see this in mice that were maintained on that high-fat, high-sugar Western diet. So in mice maintained on a control diet, we see protection from airway inflammation. But in mice maintained on a Western diet, we really don't. So this is another one of those plots where we reduce the dimensions so we can look for patterns. And I'm just going to point out 2 things and then tell you 2 things that I'm not showing you. So the first thing is that soluble corn fiber, or this fiber supplement, restructures the gut microbiome. So here we have mice given soluble fiber in the rings, mice that were given no supplement in the spheres, and we'll kind of ignore this upside down triangle, but we had another group in there, mice given a different type of fiber that shouldn't be metabolized by the gut microbiomes. So we see a shift in the microbiome in mice that were given a soluble fiber supplement. Now, what I'm not showing you is that when we just look at just the soluble fiber-fed mice, we see a difference between those that were maintained on a control diet and a Western diet. So we think that in the mice on the Western diet, we don't have a complete restructuring of that gut microbiome, and that could be why we don't see protection from airway disease. So to summarize this, we show that diet changes by increasing fiber causes shifts in the gut microbiome composition and can reduce inflammation in the sinuses of these mice that are maintained on a control diet, but not those maintained on a Western diet. So where are we going from here? We have a study funded by the Flynn Foundation to do a pilot clinical trial, and we've enrolled probably half to 2/3 of the individuals that we aim to, and we have a little bit more time on this. So this is at Phoenix Children's Hospital with Dr. Matthew Rink and Dr. James Woodward. And we're doing a double-blind placebo-controlled fiber intervention. So we're recruiting children who have asthma, and we're trying to recruit children who have that allergic type asthma, so the IL-4, 5, and 13, but whose asthma is stable, because we don't want to give kids any other type of intervention if they keep having asthma attacks. And so what we're asking them to do is for 4 weeks, every single day, take a packet, and it's either going to be fiber or placebo, pour it into orange juice or some other fruit flavored beverage, because a placebo is literally sugar and they could tell if they were drinking sugar or not, and then drink that daily for 4 weeks. And we've gotten good compliance. Kids come back and we get 80 to 90% or so compliance. And what we're asking is, does this fiber supplement change the microbiome? Do we see increase in those short chain fatty acids? And do we see any change in their inflammatory response? And so this trial is ongoing at PCH, so I do not have those data for you yet, but hopefully at a next Festival of Science, we can talk a little bit more about that. So what are the key takeaways that I want you to come out with if you have been listening to this talk? A, we are at least as much microbe as we are human. Our microbiomes are incredibly dynamic and modifiable and can be altered by environment, such as diet. Our microbiomes influence host health status and are often altered in composition or who's there, or diversity, how many of different types of microbes are there, in chronic and progressive disease. And in these complex diseases like CRS, asthma, even Alzheimer's disease, and cystic fibrosis, it's likely a complex interplay between both our gut microbiomes, our environment, and our respiratory microbiomes that contribute to health and disease. So with that, I want to thank this very big list of people, my lab who you met in that video, Greg and Andy for helping us out, our clinical collaborators both at UCSF and Mayo Children's Hospital, and then our funders, National Institutes of Health, Flynn Foundation. And I didn't talk about our Alzheimer's research, but we have been supported the last few years by the Arizona Alzheimer's Consortium. And would also thank all of the Flagstaff Festival of Science sponsors. And I'm going to go ahead and end my slideshow here, and I'm happy to take any questions.
All right. Well, that was wonderful, Emily. Thanks so much.
You're welcome.
I'm not seeing any questions right now in the Q&A. So folks, if you have any questions, now's the time. Just type it right into the Q&A box and we can ask Dr. Cope here. Oh, here's one. Could you let us know what is the future of microbiome research in the CRS and asthma?
That is a good question. And what we would like to see happen, and there are companies, for example, my former postdoc advisor has started a company that is looking to develop microbial consortium that are rationally designed, that is designed based on science in order to treat as a probiotic, so that's a live organism, or a prebiotic, which is like the fiber supplement or something that makes the good microbes grow, or some combination of the two. And so I think that these over-the-counter probiotics aren't directly targeted for specific diseases. So we have a lot of work to do in designing, sort of rationally designing these consortium to prevent or treat disease.
Awesome. Okay, we have one question from a participant who says, thank you, that was great. Can you give us a couple sentences about the Alzheimer's/gut connection?
I could give you a 45-minute talk on that. Oh, it's so interesting. So the microbes in our gut can communicate to our brain through a variety of mechanisms. They can produce metabolites. They can interact directly with the enteric or vagus nervous system, which interact with the central nervous system, or they can induce things like inflammation or reduce things like inflammation. And so in Alzheimer's disease, well, in addition, in Alzheimer's disease, we think that the microbes in the gut can produce things like LPS, or even like aggregated proteins that aggregate in your brain. Our microbes produce those too. And so we think that there are a lot of different mechanisms by which the microbes in the gut can communicate with the brain. Now, what we're seeing, and this will be my second and last sentence, I promise. What we're seeing is we have mice that model key pathologies, so plaques and these neurofibril tangles that are key in Alzheimer's disease patients, as well as neuroinflammation. And the mice that model these pathologies have differences in their gut microbiomes that come before these pathologies develop in mice. So we have a couple of grants to look at really what this means.
Well, there's a follow-up to that question. How did someone even get the idea to look at the microbiomes in connection with respiratory or brain diseases? Where did the hypothesis even come from?
Yeah, there is a lot of smart people looking at this, and it sounds a little science fiction-y. But as our understanding of our gut microbiome and the whole connection between our microbiomes and our host kind of evolved or came to light, then we started thinking about, this isn't just staying in the gut, so where is it going? And that's kind of what led to some of these hypotheses.
Okay, we have another one. Have you ever studied CP/CF together?
Giorgio, can you define CP? if CRS and CF together is what you're asking, then yeah, we are looking at both the upper and lower airways in patients with cystic fibrosis, and we see very similar microbial communities in both body sites. And interestingly, we have patients who have undergone cerebral palsy. We have not. So I will stop what I was saying. No, we haven't looked at CP together with CF, and that might be interesting.
Okay. Any implications for the big C? You know what pandemic I'm talking about.
What pandemic? Just kidding. Yes. So yes, there are probably many implications for the big C. We do have a grant application in that is asking about the relationship between the respiratory microbiome and ACE2 gene expression. So ACE2 is one of those receptors that SARS-CoV-2, the virus causing COVID-19, binds to. And we hypothesize that certain subtypes of patients with chronic rhinosinusitis have more ACE2 expression that's mediated by microbiome interactions. And so that could potentially cause people to have a higher viral load in the upper airways or potentially a greater disease burden.
So I'm going to chime in real quick because this turned out to be way more pertinent to my family than I anticipated. My sister has CRS and asthma.
Oh.
And as an adult, is your research, can I say, hey, maybe you should try a prebiotic soluble fiber in addition to your diet? Is that something that you can, I mean, I can't imagine it would hurt her to do something like that, but is that something that you could say, sure, give it a try, it can't hurt?
Yeah, I mean, I'm not a clinician, so I can't say that, but I hope that our trials will point to that. It certainly does in mice. And so I hope to have more information as our pilot trial ends, and then also we're putting in another application to put a larger trial together. And luckily we have these great collaborators at PCH. So I think pretty much anyone with kids with asthma, even here in Flagstaff, probably see Dr. Rink and his colleagues at PCH. So I do hope that we can be more definitive on that in the future.
Thanks. Sorry to barge in on that.
No.
It was a very pertinent question.
It was.
Okay, I think we have time for a few more here, a couple more minutes. Are there any fungi in our airways too? If so, do they cause CRS or asthma?
Right, so I focused on bacteria mostly here. And indeed, we have a fungal microbiome in our airways and our guts. We think that some species are commensal or even beneficial. So we see a genus called Malassezia, which is a fun one in almost everyone's noses. We also, sorry, was kind of looking at the Q&A as I was answering that. We did a study in mice where we showed that who is hanging out with Malassezia, which bacteria, different bacteria hanging out with Malassezia in the nose could actually cause increased inflammation. So it's a very dynamic community up there.
Wow, it's fascinating. All right, do we have any other questions? Did I miss any?
Yeah, I think there's a question about participant trials.
Oh, I got lost in all my things here. Yeah, participant trials, your views, are you for them?
Like clinical trials? So we are running one for asthmatics, and we're still enrolling individuals. So if anyone knows anyone who has asthma and is between 6 and 17 and is willing to eat fiber or sugar for 4 weeks, certainly talk to your folks at PCH.
Okay, well, if there are any other questions, we have about a minute left here. Otherwise, I just want to say thank you to Emily. This was fantastic. I learned a lot. And I'm looking forward to seeing if we do this, if you participate next year, what else you come out with.
Yeah, it'd be great. Thank you for having me.
Thank you so much. And we've hit 7:30. We're perfect timing. Really appreciate it. For those of you out there still hanging in there, please go to scifest.org to check out the rest of the program for the rest of the week. Thanks so much.
Sounds great. Thank you,
Good
guys.
night.